Okay, so the question is to describe photosynthesis. So photosynthesis is the process by which atmospheric CO2 and water from the soil are combined to make a simple carbohydrate, usually represented as glucose, and molecular oxygen. Now, this process is driven by light and is the basis for the vast majority of life on this planet. So this can be broken down into two broad classes of reactions. First is the light reaction. Second is a dark reaction. That's not a very good descriptor for this set of reactions for reasons that I'll get to in a minute, but for now, it's a useful way to keep them separate. So photosynthesis takes place in all green parts of the plant, stems and sepals and so on, but mostly in the leaves. And if you look at a cross-section of a leaf, you'll see cells packed closely together on the top side and more loosely on the bottom side. These are all parenchyma cells. The tightly packed ones are palisade parenchyma, and the loosely packed ones are spongy parenchyma. And then you have these pores that are actively opened and closed by the plant in response to the environment, called stomata, mainly on the underside of the leaf, but also some on the top side. The function of those pores is to allow CO2 to diffuse into the leaf and to allow water vapor and oxygen to diffuse out. So these cells, these green cells, contain organelles called chloroplasts, which are bound by a double membrane, the result of an ancient endosymbiosis when a photosynthetic bacteria was engulfed by a plant that then failed to consume it. And the key structure here are these stacks called grana, which are bound by a thylakoid membrane. They're kind of like stacks of pancakes that are connected by bridges that are also made of pancakes. And the lumen of the cell is called the stroma, and then the thylakoid membrane binds the grana. So the first part of the reaction, the light reaction, takes place on the surface of the thylakoid membrane. And that membrane is a lipid bilayer. In which is embedded a number of important proteins. I will describe approximately 5 of them in detail. So in between, you have more bilayer. This side is the stroma, and this is the thylakoid interior. So the critical light capturing devices are photosystem 2 and photosystem 1. Photosystem 1 was discovered first, photosystem 2 was discovered second, but in time, photosystem 2 takes place first. Photosystem 2 captures light from the sun at a pigment called P680 because its absorption spectra is centered at a wavelength of 680 nanometers. There's also a large number of accessory pigments that are able to capture a broader wavelength of light and then bounce it down to this wavelength, but at the reaction center, the energy is at this wavelength. And so what photosystem 2 does is participate in a reaction where molecular oxygen is split, and each molecule gives you, when water is split, and each molecule gives you half of a molecular oxygen and two protons. Now, oxygen in water is extremely oxidized. The oxygens are much closer to the oxygen than they are to the protons. And so it requires a significant amount of energy to strip those electrons from water and bounce them up to a higher energy state. So these protons, as this process continues, these protons accumulate and diffuse within the thylakoid. We'll save them for later. In the meantime, the electron, the excited electron from photosystem 2, is then delivered to the reaction center of photosystem 1 through a process that's poorly understood. There's some quantum effects involved regarding the ability of an electron to appear to be in two places at the same time or to tunnel between two locations without being at an intermediate location. But to make a long story short, in spite of the fact that these two systems are spatially separated, the electron is able to be delivered from one to the other. And the pigment in photosystem 2 absorbs at a slightly longer wavelength of 700 nanometers. Now in photosystem 2, the electron is again excited through the addition of more light at the photosystem and at the accessory pigments. And at this point, that energy is used to increase the concentration of NADPH via reduction of NADP. So this is one of the key outputs of this part of the cycle. Meanwhile, this accumulation of protons is then able to participate in the synthesis of ATP from ADP and an inorganic phosphate through the action of a proton motive force, where these protons diffuse down their concentration gradient through the ATP synthase motor and store energy in the form of ATP. So the output of this process is the reducing agent NADPH and the energy storage molecule ATP, which are then able to participate in the dark reactions. Also, as a byproduct, you get molecular oxygen, which then diffuses out of the plant and into the atmosphere and is available for use by other organisms.